Meiosis, cell division that results in the formation of sex cells. This process has to be different from mitosis because we don't want identical copies. We want half the amount of DNA so that when the sex cell is fertilized with another cell with half the amount of DNA, the product has a full complement of DNA. There are two parts to meiosis. Reduction division sees the homologous pairs separate. The once diploid cell, having chromosomes paired with their homologous other, now becomes haploid. The haploid products of meiosis I have a maternal or a paternal chromosome number 1, and no longer both. Same with the chromosome number 2 and 3, and so on. In males, one of the haploid cells will have the X chromosome, the other will have the Y chromosome. Meiosis II has products of recombination, the definition of which we'll go into later. The haploid cells of chromosome 1 still has its chromosomes as sister chromatids. Meiosis II separates these sister chromatids in much the same way as mitosis. Interphase, prior to meiotic division, is the same as for mitotic division. DNA replicates during S phase, and the cell gets bigger to accommodate the division. The first of the two divisions, meiosis I, begins with Prophase I, not necessarily the longest part of the process, but one where a lot happens. In mitosis, the identical strands of DNA supercoil into chromatids and connect at the centromere. In meiosis, not only are the sister chromatids paired, forming a chromosome, but the homologous chromosome pairs up with it. The coming together of the homologous chromosomes is called synapsis. During prophase I, synapsis occurs and the overlapping arms and legs of the homologous chromosomes called crossing over, at positions called chiasmata, pair the chromosomes as they are moved to the equatorial plate of the cell by the spindle fibers. In mitosis, human somatic cells see 46 chromosomes, paired with their identical sister, spread along the equatorial plate. In meiosis, the cells that will ultimately produce the sex cells see 23 pairs of homologous chromosomes along the equatorial plate. Note that in this diagram, showing metaphase I, the homologous pairs are arranged so that the big green one and the small blue one will be pulled to the north end of the cell when anaphase comes, and the big red one and the smaller yellow one will go south. The arrangement of these homologous pairs is random and unaffected by neighboring homologous pairs. That is to say, the blue-green combo at the north could just as easily have been a yellow-green combo. And in other meiotically dividing cells of the same individual, that could indeed be the case. With 23 homologous pairs, the combinations of which chromosomes will appear in either end of the cell during anaphase numbers in the millions, over 8 million in fact. This phenomenon contributes to the genetic diversity between the offspring of the same couple and is called independent assortment. Seen more clearly at this point are the effects of crossing over. Whole sections of DNA between homologous chromosomes are exchanged, meaning even the sister chromatids are no longer identical. The extent of the crossing over varies with the size of the chromosome. The larger chromosomes will have more chiasmata than the smaller ones. The combination of chromosomes in this cell mean the resulting cells following telophase have a green and blue at one end and a red and yellow at the other. Notice that at telophase, the resulting cells at the end of meiosis I are haploid. The mechanisms for these stages of meiosis are similar to mitosis. The second part of meiotic division, meiosis II, is more reminiscent of mitosis. Meiosis I sees the separation of homologous chromosomes. Meiosis II sees the separation of the sister chromatids. Granted, the sister chromatids are, for the most part, no longer identical thanks to crossing over, but we still refer to them as sister chromatids. From prophase II to telophase II, the haploid cells divide once again to produce a total of 4 cells. It is important to see that the resulting cells that undergo no further division, but will differentiate into, in this case sperm cells, are still haploid. How is it that when a diploid cell divides to become a haploid, that when it divides again, it's still haploid? After all, the amount of DNA in the resulting sex cell is now a quarter of the amount in the original parent cell. The very definition of a diploid cell is one that has each chromosome represented by each parent. Diploid organisms have two parents, and so will have two chromosome number 1s and two chromosome number 2s and two chromosome number 3s, and so on. Haploid cells only have half the number of parents. In diploid organisms, haploid cells only have one chromosome number 1 from one or the other parent, one chromosome number 2 from one or the other parent, and so on. After the first meiotic division, the chromosomes no longer have homologous partners and are, by definition, haploid. They are, however, still attached at the centromere to their sister. The second meiotic division, however, separates the sisters, revealing cells that still have chromosomal representation from one or the other parent, and are thus still haploid. During Prophase I, synapsis occurs, where homologous chromosomes come together forming tetrads. Think of tetra, 4, meaning 4 chromatids, 2 pairs of homologous chromosomes. At the start of Prophase I, the nuclear membrane dissolves. The chromatin supercoils to form chromatids. Identical chromatids, the result of DNA replication during S phase or interphase, pair up and connect at the centromere. Centriole pairs move to opposite poles of the cell and spindle fibers form. So far, just the same as mitosis. Then synapsis occurs. Homologous chromosomes pair up. Even the X and the Y pair up due to a region of homology between the two chromosomes. The 4 chromatids together are collectively known as tetrad. So crossing over occurs when 2 homologous chromosomes align together and link arms. Non-sister chromatids link arms and may exchange DNA, and this contributes to genetic variation among the sex cells. To better illustrate the benefits of variation crossing over offers, we'll add 2 gene positions on these homologous chromosomes. They have 2 possible alleles, each represented by upper and lower case letters. During crossing over, only the homologous chromatids link arms, and this may result in the exchange of DNA. Chromatids 1 and 4 in this illustration are not shown to be exchanging their genetic makeup. Chromatids 2 and 3, however, do. Before, there was only 2 possible genetic combinations. If a chromatid has the big A allele, it would also have the big B allele. And if a chromatid had a little A allele, it would also have a little B allele. Now there are 4 possible combinations of allele distributions between the 2 genes, and this only considers one crossing over event. Some of the larger chromosomes can see several crossing over events. Some of the smaller chromatids are physically unable to cross over with the frequency of the bigger chromosomes. Where once crossing over was thought by early scientists to be an error, crossing over is now agreed to be an important contributor to genetic diversity. In contrast to mitosis, where the sister chromatids lie along the equator of the cell, Metaphase I has the homologous pairs line up along the equator. For the meiotic cell, the red chromosome could represent the maternal chromosome and the blue the paternal chromosome. It looks like the upper chromosomes in this cell, the paternal large chromosome, is set to migrate to the same pole as the maternal small chromosome. The chromosomes could just as easily have arranged themselves so that both blue ones migrate to one pole together and both red ones migrate to the opposite pole together. This variability among meiotically dividing cells is called independent assortment. So independent assortment is the random arrangement of maternal homologs versus paternal homologs along the equatorial plate of the cell. Of the 23 chromosomes facing one pole, some will be maternal, some will be paternal. It's called independent assortment because the arrangement of one homologous pair is independent of any of the other homologous pairs. The resulting gametes will have different combinations of parental chromosomes. There are 2 to the power n combinations, where n is the haploid number. So 2 to the 23 is 8.3 million. Now combine this with the variation associated with crossing over. That's some genetic variation. So the genetic variation that exists between the sperm cells of males is just as varied between the egg cells of females. And if a couple were to have 2 kids, it's not surprising some siblings don't look related. Anaphase I, telophase I. Homologous chromosomes separate to opposite poles of the cell. and some cells bypass this stage and move to cytoplasmic division. Other cells form nuclear membranes around each cluster of uncoiling chromatids. Products are haploid cells. Each chromosome contained within is either maternal chromosome or paternal chromosome, not both. So Meiosis II has similar phases to mitosis, differentiated by Prophase II, Metaphase II, Anaphase II, and Telophase II. Anaphase II involves the separation of the sister chromatids. The daughter cells are still haploid, just like the end of Meiosis I, only this time they're not in replicated pairs. Mitosis versus meiosis. Both are types of cell division. Mitosis occurs in all cells of the body, the somatic cells, except the cells that will produce gametes. Meiosis is the type of cell division for the production of gametes. Mitosis is also referred to as conservative division, where the genetic products of the daughter cells are conserved. A diploid cell divided to become two diploid cells. A product of meiosis needs to have half the genetic information so that when it combines with the meiotic product of another organism, fertilization, the entire genome will be present. So after the first meiotic division, there is a reduction in chromosome number, hence reduction division. The second meiotic division reveals a total of 4 cells where there was once 1, and all the 4 cells are genetically different from the parental cell. When the daughter cells are genetically different from the parental cells, we call them recombinants, hence recombination division.